microbiology class on the topic heredity, chapter heredity, and the topic we are going to start today is your dihybrid cross. And before going to start the class, I hope you all people are doing well at your home and you are focused on your study. So today's class, the topic that we selected is dihybrid cross. Okay. Dihybrid cross. In the last class we are completed with the monohybrid cross, and I think you people were known now the key terms regarding doing this process or experiment or cross, that is P1 stands for parent, G for stand gametes, F1 that is first failure, cross fertilization, self-fertilization. I want to give a small hint about self-fertilization, self-cross or cross. So, if both the parents have two different sources, that is, okay, they are cross-fertilized, okay. And one case, that is, if we are going to cross among the same individual, that is self-fertilization or self-cross. We used to say self-cross, because if the individuals of same generation we are going to make cross among them, that is self-cross. So, in case of dihybrid cross, the character chosen by manual is shape of the seed, second one is color of seed. Okay, one is your shape of seed and another one is your color of the seed. Now, what is the shape of seed? Two traits are chosen. Now, we need to focus on dominating and recessive. That which one is dominating and which one is recessive. So, in this case, dominating is going to be represented by capital R. Okay, capital R, which stands for round. Okay, which stands for round. And for the recessive, we have chosen is small r. Okay, small r, which stands for wrinkle. Okay, so in the case of shape, okay, in the case of shape, dominating round and wrinkle, so recessive wrinkle. Dominating is going to be expressed by capital R, and that recessive is expressed by small r. First trait this is. Second trait is color of the seed. Okay, that is color of the seed. Now, in the case of color of the seed, one is dominating. One is dominating, that is yellow. Okay, yellow, which is represented by capital Y, and the second one is your recessive. Recessive trait or recessive in the case of color is your green. Okay, green, which is represented by small y. So you all know very well that always dominating characters or trait going to be expressed by capital letter alphabets, and small one is going to be represented by small letter. Okay. So what are the two characters chosen? One is shape of the seed, and what are the contrasting features in that? One is round and another one is wrinkle. Got it? Round one is dominating and wrinkle one is recessive. Done. This side. Another trait is color of the seed. Okay, another trait chosen by Mendel is the color of the seed. One is dominating and another one is recessive. Dominating, that is yellow, represented by capital Y, and the recessive, that is green, represented by small y. So we got the parent and their type. Okay, now we will start the cross. In the last class, I told you that Mendel chose homozygous parent. Okay, homozygous. So one is your homozygous dominating and homozygous recessive. So capital R, capital Y. That is round, yellow. One parent is your homozygous, that is P1. Okay, P1 is round yellow, and another one is your small r and small y, that is wrinkled green. Okay, wrinkled green. This one is your recessive trait. Got it? Now what will be happen? From the parents, gametes are going to be formed. So from here you will get capital R, capital Y. They are diploid in number, right? They are diploid in number currently. But when gamete formation takes place, you all know very well that reduction division, that is known as meiosis, going to take place. As a result, the chromosome number going to be reduced half. So you will get gamete like RY. Again, this is going to segregate to this one. RY again means yes, or now they will cross with each other. So this is the form what you get. Okay, the gamete type. Okay, and another one is your wrinkle, that is, and in this case, recessive case, the gamete you are going to get small r, small y, small r, small y. So this is the form that you will get here. Okay, so Mendel has given few laws, that is law of independent assortment. Okay, law of segregation, law of independent assortment, and law of dominance. So here you will see each gamete is independent to assort themselves to any of these gametes. Means gamete of one parent is independent to assort itself or gene. Okay, assort itself to any other of these gametes. So F1 generation means when these gametes going to be crossed with each other. Okay, cross with each other, the F1 individual that we are going to get, they all have the same genotype and phenotype ratio. Okay, they have the all same genotype, means genetic makeup and physical makeup. Okay, how come? See, when this is going to cross with this, you will see that capital R, small r, capital Y, small y. This is you will get in each and every cases here in the F1 generation. Got it? So let's assume that all the individuals of F1 generation are genetically and morphologically same. Why I am saying this? Because see, in both of these cases you will see the heterozygous condition. First of all, one is capital R, one is small r. Here you also see capital Y, small y, same here you can see. Okay. Now this is the genotype. What about morphology or phenotype? This is going to be round yellow because according to law of dominance, I told you that if 50% of dominating trait is going to be present, then the expression of dominating is going to be takes place, not the recessive one. Okay? So this is going to be expression of round and yellow. So in F1 generation, all individuals are heterozygous and the morphology or phenotype is round yellow. First thing, okay. So Mendel here taken two characters, that is one homozygous dominating and another one is homozygous recessive with round yellow and wrinkled green. He didn't found a single of wrinkled green in F1 generation. In F1 generation, he did not found any single of recessive trait. Okay. So what he did, he made self-cross. Okay, self-cross among the individual of F1 generation. Okay, he made self-cross among the individual of F1 generation. So now the individual of F1 generation he selected is going to be act as T P2, second parent. Okay. So just check this. What we have got the genotype, we will write here. Okay. This is the product of first parent. Okay, that is F1 generation. Now check here. P2 means the F1 individuals, those who are going to act as parent now. What you got the ratio? Capital R, small r, capital Y, small y. Yes, this is what we got. Yes. And another one also have the same genetic makeup. Now what will be happen? They are going to produce G2. They are going to produce G2, gamete 2. What you will get here? Gamete, capital R, capital Y. See, these are the diploid parents which are producing haploid gamete. So what you see, this is actually the gene present in the chromosome which is going to be halved during the process of meiosis. Okay, during the process of meiosis they are going to be halved. This is a particular homologous chromosome, this is also homologous chromosome. They are going to be half and independent to assort themselves. Don't get confused, okay? So, the gamete type that you will get is this, second one is this, and third one is small r, capital Y, and small r, small y. So these are the gametes that you get from this parent. In the same way, suppose this on this father side and from the mother side again, you will get the same pattern because F1 individual has the same genotype. Okay, capital R, capital Y, one gamete, capital R, small y, and so on. Small r, capital Y, small r, small y. So this is the gamete that you got from the P2. Now see. When we are going to make cross that for the F2 generation, okay? For the F2 generation, so simple method of using this method is not suitable because 4 gametes when crossed with alternate 4, there is probability of total 16 individual for square, that is 16. Okay, 4 into 2, that is 16. Because they are in pairs now, so 16. So to use that in a proper way, okay, a scientist that is known as Punnett, his name was Punnett. So depending on his name, he developed a square, a chart which is known as Punnett square. He made column and vertical and a horizontal, okay, and use one column for the mother and one column for the father, okay, means one is maternal gene and another one for the paternal. So we will use that, and in the column we will use the gamete on selfing. Okay, this is selfing. When we make cross among the individuals, okay, among the individual of F1 generation, that is known as selfing. So now we will draw Punnett square. Now you will write the gamete here. What you have got? Capital R, capital Y, capital R, small y, small r, capital Y, small r. Correct? This is the sequence of the gamete. Same thing same going to be here because all individuals are genetically and morphologically same, so they are going to produce same kind. Clear? Now we are going to make cross, and this gamete going to cross with all these four. Again, this one is going to cross with all those four. So in this way, we are going to fill this chart. So what you will get here is capital R and capital Y. Here also capital R, capital Y, and small y. Okay. So remember, okay, be careful while doing this cross. Capital Y, and capital Y, small y. This is what first. Okay. Then second. So this is what you will get in F2 generation, okay? This is what you will get in F2 generation. So see here, you will see that depending on the genotype, you can find out the phenotype. This is going to be round yellow. This is going to be round yellow, round yellow, round yellow. Yes, these all are round yellow. Here you will get round yellow. This one is a round green, okay? Round green because small y, homologous condition. This is also round yellow, round green. Okay, here you will get round yellow, round yellow. This is wrinkled, the two are smaller, wrinkled yellow, capital Y. Here also wrinkled yellow. Okay, here you will get round yellow, round green, wrinkle yellow, wrinkle green. So this is the genetic or phenotype you got. Now we are going to classify this as genotype and phenotype. So in your case, they are going to be asked phenotype. Okay. So round yellow, 1, 2, 3, 4, 5, 6, 7, 8, 9. You will get 9 round yellow. Okay, round yellow. How many? 9. Okay. And after that, wrinkle green. Okay, this one. Wrinkle green. 1, 2, and 3. Okay, you will get 3 wrinkle green. 3 wrinkle green, and after that wrinkle yellow. Wrinkle yellow. Also how many? 3 wrinkle yellow. Okay, 1, 2, and this is wrinkle yellow. 3. You will get it, and 1 you will get wrinkle green. Okay, wrinkle green. Good. So what is the phenotype that you will get? Ratio is 9:3:3:1. This is your what? This is your genotype. Oh, sorry, phenotype means appearance of the plant. 9 plant you will see they are what type? They will be round yellow. 3 plants you will get wrinkle green, 3 plants you will get wrinkle yellow, and 1 you will get the recessive, homozygous recessive, that is wrinkle green. So just think, Mendel have chosen only 2 types, wrinkle green, round yellow, dominating. But what he got in his F2 generation is 4 type of variation. How many? 4 type of variation. Thus, it is law of independent assortment that gene can be independently assort themselves or segregate themselves. Got it? So this is your phenotype ratio. In the case of genotype, you can say that is homework for you. Just check this makeup. How many you are getting? This pure form, homozygous. Here you will get one here you will get. Okay. And other you will have get the heterozygous condition, but you have to find out that how many of them are having the same pattern. Okay, like this. You can see this one. Okay, this one. They are having the same pattern. Okay. So you have to find such and this one. Yes, this one going to be same. So that in this way you will get and find the genotype. This one. Okay, this one you will get the genetic type. So in this way 1:2, 2:2, 1:2. In this way you will get the ratio. Total, while you are going to add them, you will get the 16. Okay, see 9, 10, 3, 16, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. Total 16. So genotype also when you are going to mention 1:2, 1:2, 3:4, 1. In this way you will get them also what? Total 16 types. So that is going to be homework for you, and hope you people are clear with dihybrid cross. Yes. So this is what how we can use unit square for large cases. Okay, such kind of cases in the dihybrid or trihybrid cross, because general method is not applicable in that case. So this is you. Got? The next class we will discuss about how actually what is the biological mechanisms or the molecular mechanisms taking place behind the expression of all those things, role of hormones, or what is the role of actually DNA or gene, okay, while expressing this trait. Okay, so thank you, have a nice day, and yes, again if you have any doubt please leave a comment in the comment section. Thank you.